Lipids, carbohydrates, and proteins are essential to the function of all living organisms. Yet there are still a lot of unknowns about these macromolecules. For example, how do intrinsically disordered regions contribute to protein function? What are the mechanisms guiding the diversity in protein folding? DNA is the code of our lives, and unlike these macromolecules, we know how it operates. We know that DNA forms a double helix. We know that the entire code comes from four base pairs: adenine, guanine, cytosine, and thymine. Similar to a micelle, these nonpolar hydrophobic bases are surrounded by a hydrophilic backbone composed of a sugar and a phosphate group. This entropically favorable force, known as the hydrophobic effect, minimizes the surface area of water contacting the hydrophobic bases. This causes the DNA to fold in a way that pushes the hydrophilic backbone to interact with the water, shielding the hydrophobic region. This is the main driving force behind holding DNA, the double helical structure. Because we have defined understanding about DNA sequence and structure, we are able to manipulate it with some really cool applications.
One really awesome application is DNA origami, and it's exactly what it sounds like. Up until now, we're used to seeing DNA as a double helix between two complementary single strands, but by strategically using sequence and the spontaneous folding driven by the hydrophobic effect, we can synthetically develop 2 and 3-dimensional shapes from DNA on the nanoscale. First, in lab, we need to isolate a single strand of DNA with a known sequence. We call this single strand of DNA the DNA scaffold, which is somewhere around 7,000 base pairs that will eventually fold to make our intended shape. Since we know the sequence of the scaffold, we can design smaller complementary strands called staples in blue. We see here that these staples bind to the DNA, which then causes a crimp in these sections, and thereafter a chain of folding events similar to what we see in the protein funnel. Since the shape is encoded in the scaffold, researchers are able to direct the assembly. They simply develop a DNA blueprint and vary the type and number of staples to drive the folding. The staples are approximately 30 to 50 base pair oligonucleotides, and we add around 200 staples to drive the folding reaction. In a small drop of solution, this can occur hundreds of thousands of times, almost instantaneously when the scaffold and staples are present. We have seen researchers develop an infinite number of shapes like stars and smiley faces, and now we are focused on the biological applications.
Since its inception in 2006, researchers have refined the DNA origami methodology and are now exploring it in the field of bio-nanotechnology. Drug delivery is one of the most promising applications of DNA origami. The 3D origami structures, such as cubes, can encase therapeutic agents prior to injection in the desired cells. Then, the origami can be opened by several mechanisms to release the drug, potentially allowing us to increase drug concentration delivered directly to the cells. Interestingly, the triangular shapes have shown promise in targeting the cancerous cells. Some researchers have even used the origami to create DNA nanorobots capable of holding gold and silver nanoparticles and of moving. The functionality of DNA self-assembly is indeed very versatile, even allowing for DNA-protein hybrid assemblies. DNA origami can hold macromolecules and provide us with a 3D structural analysis, surpassing the limitations of current microscopy techniques. Despite these advances in spatially arranging proteins, the in situ assembly of proteins continues to pose quite the challenge. Next time, we'll see how researchers have overcome this. In our paper, they use RNA from the tobacco mosaic virus, or TMV, and its coding protein to facilitate the assembly of hybrid DNA origami protein nanoarchitectures in situ.